first I want to introduce you guys to my two study species. So on top here you have the black-capped chickadee, and the bottom the Carolina chickadee. The black-capped chickadee occurs in the kind of the northern half of Missouri. There's kind of a diagonal zone that I'll talk more about later where the two ranges meet. That kind of stretches from around midway down the border with Kansas and then intersects here in St. Louis. And so the black-cap occurs north of that, and the Carolina chickadee occurs to the south. So who here is familiar with the one or both of these species, the chickadees? A lot of people, yeah. That's part of one of the reasons why I want to study these guys. They're kind of a fan favorite, and they have characteristics that make them really easy to study. They're really curious, they're pretty social, they like to come to bird feeders, and they use nest cavities. So it's not like I have to go trekking into the deepest forests of Ozark National Scenic Riverways to find my study species. They're just right there in yards and campgrounds, which makes it really nice, especially for a 2 years master's thesis where I don't have a whole lot of time. So while those things are really fantastic about chickadees, they're not unique to chickadees. There are a lot of different songbirds that have those same characteristics, that are friendly and sociable and easy to find. So what I'm really interested in is what I just mentioned about the chickadees, is that they have this zone of contact where the two ranges come together that runs through the middle of Missouri and then kind of jogs south here. And within that zone, the two species will hybridize. So are you guys familiar with what hybridization is? Pretty much, yeah. So it's when two species will mate with each other and they'll produce offspring that have ancestry from one side and ancestry from the other side. So the black-capped and Carolina chickadees will mate with each other and they'll have offspring that are neither. They're a combination of the two. And that happens here in this zone. And what's been found with these hybrid birds is that since they have this combination of genes that don't quite match together well, they're lower in what we call fitness, which is basically just the ability to persist. So these birds are not as fertile, so they'll have fewer offspring, or their offspring will be either unhealthy or maybe they'll be stillborn. And they just they don't do as well in general. They're just not as good at being birds as the pure types. So this is why we see this zone here, where it's just very narrow and both the species can't expand into the other one's range, because the hybrids don't do well. So it keeps the Carolinas from advancing farther north, and it keeps the black-caps from advancing farther south, and the hybrid zone stays really narrow there in the middle. And I don't know if you guys noticed this little V shape down here in the east. So that is right along the Appalachian mountain range, the Smoky Mountains. So the reason for that is that part of the limitation of the Carolina chickadees range is based on climate, based on temperature. So there in the mountains, the black-caps are able to persist farther south because the climate is a lot colder with a higher elevation. So that's why you have that there. Okay, so I'm interested in hybridization, specifically in chickadees. So there has been quite a lot of research done on bird hybridization in other species as well as in chickadees. And some of my favorite examples here are this one right here. It's a paper by Toes et al. From 2016. So what they did was they went and they sampled a whole bunch of blue-winged warblers, golden-winged warblers, and then their hybrid morphs that each produce, which are named Brewster's and Lawrence's warblers. These birds hybridize like crazy, and they produce hybrid offspring that do really well, and they're pretty fertile, and they don't have as much of that fitness cost as the black-cap and Carolina hybrids do. So they sampled these birds, and what they found out when they sequenced the full genomes of both species is that there are only 6 regions on this massive genome of both of these birds. There are only 6 regions that differ between the two species, and all of those regions are related to plumage. So that kind of raises the question of, are these birds actually different species if they're that similar genetically? Which people have always said, yeah, they have to be, because look how different they look. Like up here you can see the laser pointer. The blue-winged warbler and the golden-winged warbler don't look really anything alike. So we've kind of just based our idea of species off of the appearance without really looking at the genes. And And then I have to give the nod to my advisor here. He did research on Afrotropical sunbird species in Eastern Africa. Jay McEntee is his name. That's his paper, the title of his paper there. These two species that he studied are very, very closely related. They separated into different species, not relatively speaking, not that long ago. It's still quite a long time compared to like what humans normally think about. But diverged really recently. They share really, really similar ecological space, but their songs are extremely different. So he went and he did a lot of acoustic sampling, so he recorded a lot of song. You can see here the two species that he studied, Nectarinia moroi and Nectarinia fuliborni. And then the song of the first of C here is C here in A, and then the song of D you can see here in B. So these two visualizations here are called spectrograms. So I'm going to go more into how to look at these and how to read them later. But just know that these are kind of depictions of these two bird songs. And you can see that one's quite a lot shorter than the other, quite a lot less complex. So what he ended up finding out was that even though these two birds are really similar genetically and they share a lot of the same space, their songs have diverged as a result of competition. So it benefited the species more to diverge in song so they could have better recognition of each other in the same ecological space here. So these are just two of many, many, many examples of studies of hybridization in birds. So then the question becomes, how is my research going to build off of that but also be different than what people have already done? So what I want to do is use this chickadee hybrid zone to help us understand what shapes the development of song in these two closely related songbirds. So the question for me is the song genetic? Is it learned? Or is it a combination of the two? So that's what I'm trying to look at here. So now zooming in on specifically the song of these birds. So back to the same picture here, you can see how similar these two species look just from looking at this. There are a few little things that you can look for. The black-capped chickadee tends to be just a tiny, tiny, like fractions of a millimeter on average bigger than the Carolina chickadee, which in the field, if you're looking with binoculars, you're not going to be able to tell. The black-capped chickadee has like more white on the edges of his these feathers here, which are called the coverts, and then these here, which are called the secondary flight feathers, than the Carolina, which you can kind of see. And even this depiction is pretty exaggerated. In the field, it's not quite that distinct. The black-capped chickadee tends to have a messier black to white bib line here than the Carolina chickadee. You can see that line's a little bit cleaner. And then also, lastly, the black-capped chickadee tends to be a little buffier here on the flanks than the Carolina chickadee. But these are all really, really subtle differences visually. On the other hand, if you're able to listen to these two species well into their respective ranges where their ranges don't meet, like up and way north of Missouri and down way south, you'll see that their songs are extremely different and pretty easy to tell apart in the field. So I have recordings here of each. So this top one here is the black-capped chickadee. It makes a two-note sound that people say sounds like Phoebe, and there will be a little, usually a little break in the second note, or sometimes even people consider it 3 separate notes. So I'll see if this will play here. You guys hear that? It's kind of faint.
Mm-hmm.
But yeah, it does sound like Phoebe. And then the Carolina chickadee, their songs are a lot more variable than the black-capped. The black-capped pretty much just sings that and that's it. Sometimes it'll be a little higher pitched or lower pitched, but it's the same structure. The Carolina chickadee will sing normally a 4-note song, but it can be up to 6 sometimes. And usually the structure is a high note, low note, high note, low note. It'll sound something like this. But it's a lot squeakier, you can hear. So I wonder if I can, I don't think it's gonna let me go back and play it. But you can kind of tell, if you were in the field and you heard that, you could say black-capped and you could say Carolina for the lower one, and it wouldn't be nearly as difficult than if you were looking at the two. But when you get into the hybrid zone where the two species connect, things get very weird. It's not letting me, there we go. Things get weird. You see all these different crazy songs because of the learning between the two species. So I'm gonna go see see if this thing will let me play all these songs. So they're not going to play in order. So I'm going to point out to you which song is playing when it's playing. And also, I did promise to explain spectrograms to you, so I'll do that now. So who here has seen or is familiar at all with what a spectrogram is? Anybody? Kind of, sort of, maybe? Okay, so basically what you have with a spectrogram is you can use some kind of computer software. You can import your sounds. And it'll give you a depiction of sound, and then so it'll have here on the y-axis, this is the frequency, so high, really, really low pitched to really, really high pitched. And then here on the x-axis, you have time, so the very beginning of the song, and then it plays over about 5.5 seconds. And then you can kind of see here, like this is a higher note than this one because it's higher on the y-axis, and then you just go along through time. So I'm going to go ahead and play these songs so that you guys can see how different they are from the stereotypical black-capped in Carolina. So the first one we play will be this one on the bottom here, which is really crazy. I've never heard in my life a chickadee that would do something like that outside of the hybrid zone. Okay, and then, so that, these are really quiet. I wonder if I can, I'm going to try to turn this up a little bit. Okay. So that song that you just heard was this one here. A little squeaky note, and then a straight note, and then the same again after. So that was this song right here, which sounds kind of sort of like a normal Carolina song, except for that third note, instead of being a high, kind of pure toned whistle, it's a little squeak. So that was kind of like a double black cap song. So that's one bird singing that back to back. So that's this one right here. And then that is almost a normal black cap song. But you might have noticed that in the second note here, instead of having the bee where it has the break in the second note, it just went for a normal whistle. Okay, so that was this one right here. It was probably really hard to catch, but there was that little squeak in front of the normal bee, which would be unusual for either species. That was kind of a typical black cap song. You can see that here, except it added another note on the end. So that was pretty much a normal Carolina chickadee song. Like I said, they can do anywhere from 4 to 6 notes. So that was pretty stereotypical Carolina. So that was the weirdness of the hybrid zone song. So you can see now why people have been really interested in birds in the hybrid zone. Things get really weird and people want to know why. So now I'm going to talk a little bit about what people have already done with the black-capped and Carolina chickadees with their song. So there are a few guys who have done some research in the hybrid zone, and kind of what they've come to the conclusion of, kind of reached the consensus that genetics doesn't really play a part in the development of the song in the black-capped and Carolina chickadees, because they've seen both in the field and in the lab that these birds can just learn each other's songs really freely. They're really good at learning each other's songs, so they kind of just were like, yeah, I don't really think genetics plays a part in this at all. But there are a couple limitations to these studies that I want to hopefully address with my work. So the first two studies I listed here, Braun and Robbins, and then Sadler. So these guys studied free-ranging wild birds, and they studied the song, and they did genetic sampling like I'm doing, but they didn't really make an effort to capture the full repertoires of what each bird could sing. So they would go in the field and they would record a bird for maybe 5 minutes, maybe 10. They get what seemed like a decent amount of song from it, but both of them acknowledged in their papers that what they got wasn't necessarily representative of every single type of song these birds could sing. And then this third study here with Cruzman Albano in 1995, that was performed in a lab setting. So what they did was they took nestling birds, they brought them to the lab and they hand reared them, and then they housed them separately, and they tape tutored them with black-capped and Carolina chickadee songs. So some black caps were tutored with black cap songs, some black caps with Carolina, So Carolinas with Carolina and some Carolinas with black-capped, and then they watched song development, which seems like a really great way to investigate how genes contribute to song, because then you're taking birds out of the social environment so they can't learn socially like they normally would. So you can kind of hopefully get a better idea of what genes are doing. But what I really want to focus on is looking at how the tendencies of these birds look even when they are still in the social setting. But I can do genetic sampling, I can study these birds in the wild, and I can see even when they have this social environment around, what tendencies stick, what do they still tend to do, and how can genetics be affecting that. And then the second limitation here is the way that they classify the song types and the way that they measured them. So both of these two first studies that studied the free-ranging birds in the wild, they classified song type just really simply, just how many notes does it have? Is it 2? Is it 4? Is it somewhere in between? Or they maybe used measurements of frequency, maybe measurements of order, but they kind of stuck with those. They just looked at how things looked on a spectrogram, and they didn't really think outside of that too much. So what I want to do is try to find some other characteristic of song. What I've settled on for now is, what about repertoire? So maybe one species of bird can learn many different types of song, and the other species can only learn 1 or 2. And there could be a genetic correlate to that. So that's what I want to investigate, kind of thinking outside the box with studying song. So here is my current hypothesis. I think that there are certain characteristics of song in black-capped and Carolina chickadees that are under genetic constraint. Primarily I'm focusing on repertoire, or how many different types a bird can sing. And my prediction is that when I gather all this genetic data and all this acoustic data, and I analyze, I will find a significant correlation between genotype and certain properties of song, namely repertoire. Okay, so that was kind of the first half of my presentation. Now we're going to move into what I find the most fun, which is talking about methods and how I did things in the field and how I'm planning to do things in the lab whenever I'm done collecting data. So step 1, which is really kind of step 0, because I wasn't really collecting data or anything yet, was just going up, because I study in Springfield, which is well into Carolina chickadee range. So going up north and just trying to find the hybrid zone. Because although it has been pretty well studied in West and Central Missouri, there's still a lot of uncertainty about where exactly this zone is. And as well as that, it has moved in the last 30 or so years that's been studied. It's moved north as a result of climate change. So my first step was just finding the zone. So I pretty much spent the summer either being trained on how to misnet birds by one of my lab mates, or just driving in my car and pulling off on random roads and trying to find birds that sang either mixed song, which is one bird singing Carolina song and also black-capped song, or what we call aberrant song, which is just a song that's weird and it doesn't really fit into either category. So I was kind of just like driving around like a crazy person all summer. I was like pulling off on the sides of roads and going into random parks and just like hoping that no one would like yell at me for trespassing accidentally anywhere. But I ended up finding actually quite a bit of mixed song, kind of south of Clinton, Missouri. Does anyone know where that is? Kind of around Clinton and just south of there. I had a few times where I found a bird. I was like, oh, it's singing weird song, or it's singing both black-capped and Carolina song. And so this is a picture here of me camping in Truman State Park, which turned out to not be in the hybrid zone. It was south of the hybrid zone. But I was camping, trying to listen and find some mixed song. And also just a disclaimer, the presence of mixed song does not necessarily mean I'm in the hybrid zone. It's just a good sign. You can't know for sure until you do the genetic analyses and you look at the actual genotypes of the birds. So what I ended up settling on was, if you look up here on this map, this zoomed in map of my earlier hybrid zone map, that little purple circle is the area that I kind of settled on, where I was like, okay, this is probably in the zone and this is a really easy, good, accessible spot for me. I ended up doing most of my research here at Sparrowfoot Park, which is just south of Clinton, Missouri. just off of Highway 13. And you can see here that it's kind of like nestled on this little peninsula, and it's surrounded on 3 sides by Truman Lake. So I think this kind of has a little like happy accident benefit of preventing these chickadees from dispersing to the north and south too much. So I'm guessing that the hybrid zone is maybe kind of trapped there in Sparrowfoot Park. I've heard tons of hybrid or like weird mixed song, aberrant song in Sparrowfoot Park. There are chickadees everywhere there. It's almost never crowded. The management is really nice. It's just kind of an ideal study space. So I've spent most of my time collecting data there in Sparrowfoot. So step 2 of the research process is making captures of chickadees. So the way that I do that, you can see me here. This is actually not in the hybrid zone. This is over the summer when I was training on mist netting. So this is a Carolina chickadee down near Ozark, Missouri at one of our field sites. So what we do is we set up these nets called mist nets. We set them up on bamboo poles. They're really, really fine, thin nets. They kind of billow out in these pockets. They're really, really hard to see for us, much less for the birds who are territorial and scared. And we will set up a Bluetooth speaker kind of close to the nets, and we'll play songs of the birds we're trying to capture. And then they will come in and act territorial and be like, who is in my space? And they'll fly into the nets. And then you have to extract the birds, and then you can take whatever data you need, which in my case is these 2 things. So the first thing that I always do when I capture a chickadee before I do anything else, the first thing I do is to put on 4 bands on his feet here. And I prepare beforehand, before I go out in the field, different color combinations of bands that I can use. So they have like a 3-color code. And then the 4th band is an aluminum band that has a number on it. So that number gets sent off to USGS. They keep track of every bird that's been banned in the United States, which is, we legally have to do that. And then the second thing that I do is I take a blood sample. So this is a box with all of my current blood samples from all my chickadees in it. So the process with that is, it's the scariest part, and I don't really like doing it because I'm always afraid for the bird, but you just have to like extend the wing out. You take a hypodermic needle and you just stick the ulnar vein, take a capillary tube, and you take a little bit of blood. And then you deposit the blood into one of these little things that has lysis buffer solution in it that keeps the DNA from degrading. So those are the 2 main things. Once I get those 2 things, I can breathe and don't have to worry as much. And then I also will take morphological measurements from each bird. So those will be measurements of wing, tail, the tarsus, which is the long foot bone that birds have, and then weight, and then some bill measurements. So this is me measuring a chickadee in the field a couple weekends ago. This is one of our undergrads in the lab, Gabriella. In these measurements, I am not sure exactly if I'll use them or not. It's just good practice to have morphological measurements of your study species. Some people actually do use the ratio. This is such a like specific little thing, but they'll use the ratio between the wing cord length and the tail length to tell apart black-capped and Carolina chickadees. Which obviously, if you're looking at them with binoculars in the field, that's never ever going to be useful to you. It's only useful if you can have a bird in hand. So that's morphological measurements. And then step 3, which has to come after the first part, because the birds have to be color banded, is the recording process. So I did a lot of my captures back in the fall. I'm still actually continuing with captures because field work is crazy, things go wrong. And I am going to be continuing with captures through the end of this semester. But then the next thing is that you have to come back and you re-find your captured birds and you record their song. And what I'm trying to do is I'm really, really focusing on trying to get the full repertoire of these birds. So I'll go out, I'll pick a bird to focus on for the day that's banded and bled. And I'll record him as long as possible, follow him around. Usually I'll have someone with me to help me spot, to look for the color bands, because when you're holding a mic, like you can see here, I use a parabolic dish-mounted microphone, and when you're holding that mic, it's really hard to follow a bird. So I have a spotter with me. I'll usually come back and record this bird on multiple separate days. in case he's getting stuck on one song type for a day. So I want to make sure I really have the full repertoire. And actually, I have found that it is much easier to make a capture in the spring and leave the bird abandoned and then just come back like the next day, because he won't hardly have moved at all. It's much easier to do that than it is to do captures in the fall and then like come back in the spring and be like, did he move? I don't know. Like I actually lost most of my fall birds that way. But so that is the third part, and I'm still doing that, and I'll probably continue through most of the summer with making recordings. Okay, so first, does anyone have any questions about field work? Yeah.
Do you have a better chance of getting your song recording early in the morning or in the evening? Which is better?
That's a good question. We usually try to be out at dawn because chickadees will. There's a dawn chorus which has like all birds like going around being territorial, singing on their perches and their territories. So chickadees will usually sing more at dawn like most other birds. The only thing that makes it hard is that everyone is singing at dawn, so you end up with these recordings that have a lot of noise from other birds. But I usually try to get out there at dawn, and I'll just stay until the bird isn't really singing much anymore, which is usually like mid-afternoon or so. But yeah, that was a good question. Any other fieldwork related methodological questions? Okay. So that's what I've done so far and what I'm still doing and probably going to be doing like well in the summer. I definitely plan on being done like probably midway through the spring semester with all the fieldwork, but just, yeah, if any of you are planning on going into fieldwork, make sure you build in extra time for things to go wrong, because they will inevitably, you'll have to work through that. It's very character building. So lab work is going to be my next step. That'll be coming this summer, hopefully soon. So once I have all this data collected from the field, it's great that I will have it. I'll have it all there with me. I'll have all these recordings, I'll have all this blood, but it's not doing anything. It's just sitting there, and I need to extract information from it. So for recordings, the way I do that is that I use this software called Raven Pro to make spectrograms, which the same program to make this I made those spectrograms with that were earlier in the presentation. So I'll load those here. I can see the spectrograms, and then basically you just like, it's really easy to use. You click and you draw a box around each chunk of song. So these should be numbered 1 through 4, but I guess I'd already done a measurement like on a different song. So you can see this is the box. It might be kind of tough to see, but can you guys see that there's a box around this first note here? A line through the middle. So you draw a box around each note. And then the program takes all this information out for you. It'll tell you how long that note is. It'll tell you what the highest frequency and lowest frequency of the note is, using this peak frequency trace that you can see this green line through the middle of each note. So that basically takes like the loudest frequency of that note and it traces it. So it gives you all this information that you can use later in analyses. So even though I don't really know for sure what characteristics of song I'm going to be studying yet, I have all these measurements and I can figure that out later. And then for the blood, what I'm going to be doing in the lab is I'll perform DNA extractions on each blood sample. So that's just basically going to involve a bunch of lab work with a bunch of different reagents and a bunch of like me pipetting things for hours. And which is not my favorite work, but it's necessary, so it's fine. And then I'll send all of those extracts off to an external lab to be sequenced. You can do sequencing yourself, but it requires a lot of knowledge of molecular methods that I don't really have, and that I'm more of a field biologist, so I'm going to leave that to other people. So they're going to do my sequencing for me, but I'll still have to do all my extractions. So that's what's coming, and now for kind of what I have so far. So my goal for sampling from the hybrid zone is that I'll get 10 chickadees. Oh, it should say from the hybrid zone, just pretend it does. I should say 10 chickadees from the hybrid zone. So 8 equals 10 means I have 10 birds that I have banded, I have blood samples from them, and I have really thorough recording of all those birds. It doesn't seem like a whole lot, but when you think of the amount of work that goes into each one of these data points, it's just very time consuming. And then I plan to have that same process done for 4 pure black-capped chickadees and 4 pure Carolina chickadees. So I will go far up into the ranges of each of those to make sure I'm getting pure types. And I will do that same process. And this is what I currently have. Right now I have I've done 6 chickadees from the hybrid zone. I haven't done any pure types yet. So you can see that I still have a lot of work left to do. But I have at least gone through the process of figuring out what works and what doesn't work. So I'm hoping that things go quicker from here on out. So, and then here I just included a little data table with what I have so far. So I have a couple of birds, one of them that I bled and banded and then he disappeared and I haven't seen him. So I don't know if he passed away or if he moved somewhere else or what. Then I have one bird that I have resighted that I sampled in the fall, and then I've recorded just a little bit, but not all of them yet. I did name my birds. You're not really supposed to do that, and I didn't find out that you're not supposed to do that until I'd already started naming them, and it was too late. So I just kept naming them. But just for future reference, my advisor let me know. He was like, "Yeah, you really should name your study organisms. It's just not a good idea." So just be warned about that. And then these are their color band combinations. The 1 or 2 letter abbreviations for each color. And then these are their locations where I've been recording and sampling them. So that's just a little bit of preliminary data there. So now kind of for what I've learned so far from this whole process, because I don't have really concrete results to show you guys, but I can kind of tell you what I've observed in the field and what I've learned about these birds. So first thing is that the areas that I'm sampling, which is primarily Sparrowfoot Park, have a lot of mixed and unusual song. Every single bird that I've used as a data point and have recorded has sang Carolina chickadee song and black-capped chickadee song and weird song, and it sings all those different types pretty frequently. And that is actually, I've been finding out from reading the literature, that's kind of unusual. A lot of studies that have been conducted in the hybrid zone have found maybe a little bit of mixed singing, but most birds singing one song type or another, and maybe they'd have like 3 or so song types total. But having these birds that'll sing like upwards of 8 to 10 to 12 different song types is pretty unusual from what I've been reading so far. So that's one thing. And then the other thing is that chickadees in the hybrid zone are weirdly not that territorial. Something I noticed pretty early on. Will be following these birds around, recording them, and I'll have 2 male birds that are like singing from the same tree, and they're not acting aggressive at all. They're just singing and minding their own business. And that's really unusual for songbirds, because normally they have territories that they hold and defend. And if another male enters their territory, they'll fight, or they'll at least act like they're gonna fight. And these birds don't really seem to do that, so that's an interesting thing that I've noticed about the hybrid zone, which kind of makes me think about future questions that my study could bring about. Like after I'm done, other work could be done in this area of the hybrid zone. I think it would be interesting to see an investigation on what are the factors that affect how many different song types develop in different parts of the hybrid zone. Because there are some parts where there are just a couple song types, and some parts like Sparrowfoot where there are like 10 to 12 different song types. And then something else could be the differences in territoriality between pure black-capped chickadees and Carolina chickadees versus the hybrids, to see if there really is a significant difference in how territorial they are, and then maybe a follow-up to that, what is causing this difference? Is this something to do with song? Song not functioning as well in the hybrid zone as it does outside, or is this something completely unrelated to song? So even though I don't have results yet, I have a lot of thoughts and I have ideas bouncing around. But yeah, I mostly wanted to talk to you guys about methods and about background and about why I find this stuff so cool. So that is all that I have. Thank you so much, and I would love to take any questions that you guys have about my research.
I have a question for you, Shelby.
Uh-huh.
Why chickadees, and why not name your chickadees?
Oh gosh. So why chickadees as in why did I study them?
Yes.
Over? I like that people like them because I like to talk to people about my research. So it's cool when you study something that people like to talk about or they can relate to. Like I was talking to like an art professor at, like way outside my department, and she was getting excited about it. She's like, oh, I see chickadees in my feeder and stuff. So it's nice because I can relate to many different people. And then why not name them? So there are a couple reasons. The first one is if you're a person who can get attached to things and you name your study organisms and then they like pass away or something happens that's sad, it can be a bummer, which is kind of more minor. And then the other thing is that if you're studying behavior, which isn't as much of a concern with my study, but if you're doing a behavioral study and you name, like, for example, one of my birds I named Frank Ocean after the singer. And Frank Ocean has made incredible music, and then he hasn't made music for quite a long time. So you can start attributing these characteristics of real people to the organisms you're studying, and kind of make correlations that aren't really there if you name them after real people. So it's kind of, it's not like the end of the world or anything, but it's just cautionary, really. Yeah. Mm-hmm.
I noticed that your Frank Ocean bird was marked as MIA. Was that before or after Frank Ocean, the singer, went MIA?
That was after. And I had to laugh a little bit because I named him when I first captured him because I was having a Frank Ocean phase, which I'm still in. He's great. But then afterwards I was like, of course he would disappear. Of course he would do that to me.
That's so like him.
Yeah, and it really is. It really is.
So I have a question. You mentioned the golden winged warbler, living warbler, their plumage. Has anybody looked at the genetics of their song? Because they do have different songs.
Yeah, they do. They have, their songs are also different. I'm not sure if anyone has done a study like that where they've done acoustic and genetic sampling on
Or
it.
any other birds that they've done the acoustic genetics.
Yeah, well, my advisor did that on the African sunbirds, and there have been others too. I think back when the eastern and spotted towhee were considered rufous-sided, I think there was some work done with theirs because their songs are also a little bit different. So, but yeah, I think more work needs to be done combining vocal and genetic stuff because a lot of people will look at like plumage and because it's much easier. You don't have to like record a bird for days. But,
Do you recapture and unband them, or they just stay banded?
They stay banded forever.
Forever.
Yep, it's for life.
Yeah.
I guess I had a question about, maybe I'm asking you to look into the future about what your results might be, but you mentioned the location of the park on a peninsula. Do you think that has anything to do with the large variety of song?
Yeah, I definitely think it could have to do. I think my kind of guess is that it's possible that the hybrid zone is kind of stuck there because chickadees do not like to disperse over bodies of water. I think that someone studied that at one point, that they'll like try and then they'll like go back, or they just won't even try at all. So I think it's possible that the hybrid zone, although it is moving, kind of stuck there, and then the hybrid birds have learned from each other, and pure types have contributed a little bit, and they just have this crazy mix of stuff instead of the zone just moving and then leaving a little trail of weird song, but kind of just steadily moving. I think it definitely could have to do with it. Yeah.
So I'm a geneticist. You say you're going to send your samples off for genotyping. What technique are you? Or
So right now we're thinking about RADseq.
Okay.
I'm not sure exactly because I know there are options and options of RADseq. You can like look at double digest, different kinds of things. But yeah, we're going RADseq. I was actually planning on doing a genome-wide association study when I first started, trying to like do RADseq, then do a GWAS, and like try to use that to find phenotypes that.
For a master's degree?
Yeah, that's why we decided not to, because I was not going to get nearly the depth of sampling I needed for that, and it was going to take way too long. So, but maybe for a PhD, possibly something like that. Yeah.
So, sorry.
No, go ahead.
Yeah, no, go on. I'll let you.
Okay. All of your hybrid individuals, is there an assumption that they're F1s?
So probably a lot of them are, because the F1s are not super fertile. So I think there are a lot of F1s and a lot of back crosses. I don't know that there are very many F2s or similar. But yeah, I think a lot are going to be F1s, and then the F1s will mate with pure types, and they can produce offspring. Good questions.
When you see the two birds singing in the same tree, are their songs similar or different?
A lot of times they will, when they're doing that, they'll be singing. I mean, I've heard both. I can remember two separate instances in my mind. One where they were both singing the same song, and one where one was singing a kind of modified black cap song, and the other was singing a more Carolina type. But they don't seem, another thing that I should have mentioned is, a lot of times, Carolina chickadees, since they have different song types they can use, they'll do this thing called counter singing, where they will try to match the song that their neighbor's singing. I didn't see a lot of that in the hybrid zone either. They kind of just seemed to stick with whatever song they were already singing, and they didn't really counter sing much. That's a good question.
So I know you mentioned about the morphological measurements for the black cap versus Carolina birds, that they're relatively similar. So it's, you know, unless you have it, you can't sort of get
Right.
a good data out of it. But does that mean that like the anatomy of these two birds is also similar to like their singing organs that they have that
That's-
contributes at all to their song type?
That's a really good question. There's a guy who works out at University of Colorado in Boulder who does more of that kind of like anatomical, like he's looked at the brains even, and I think that he's looked at vocal organs, and I think that he's found that they're pretty similar. So I think that the thought right now is that it's something neurological.
Gotcha.
If there is a genetic correlate, that it's something probably neurological.
Thanks.
Difference. Yeah, good question. Anybody else? Well, thank you so much, guys. I enjoyed talking about this quite a lot. Yeah, this is great. Great to have you. Thank you.
Good job, Shelby.